Thanks everyone for joining us today for DataBytes with the National Consortium for Data Science. My name is Amanda Miller, and I'm the Associate Director for the National Consortium for Data Science, also known as NCDS. We are thrilled to host Navarun Dasgupta and Anuja Panthari to present DataBytes, Data Viz, Chemistry, Art, and Public Health, Making Sense of Street Drugs. I'm certainly part of the Navarun Dasgupta fan club, and by the end of this webinar, I have a strong suspicion that you will be too. He has been on numerous podcasts and has speaking engagements internationally on a regular basis. But we are just delighted that he is going to be presenting with Anuja this afternoon for us. So thank you so much, you two. And before we begin, I'm going to share a few housekeeping items. Just so you all know, we have disabled the Zoom Q&A for attendees, and chat is also disabled to allow Nabarun and Anuja to present without distractions. But we do offer a place for you to share your comments and suggestions and questions. Those should be uploaded to our Easy Retro board. It's our preferred crowdsourcing Q&A platform. And we're going to go ahead and pop a link in the Zoom chat now. And right before we get to the Q&A portion of this event, we will be sharing instructions on how to use that. It's fairly easy to use, but we want to make sure everyone has the opportunity to ask questions if they have them. So if we don't get to your question during this event, we will do our best to address it in wrap-up materials. So without further delay, I'm going to give the mic to our speakers and take it away, guys.
Thank you so much for having us, and thank you everybody for sharing your lunchtime or morning time with us. So if you could go to the next slide. So we work at the Opioid Data Lab, which is a collaboration between the University of North Carolina, along with the University of Kentucky and the University of Florida. Next slide, please. The studies at the Opioid Data Lab encompass three domains: theory, practice, and lived experience. So theory studies are basic biostats, epi methods, psychology studies, and pharmacology. The applied studies in practice are really looking at the practice of medicine, the practice of pharmacy, how vital statistics are calculated, how we measure overdose deaths in the United States. The lived experience studies are applied and practical. They are initiated by people from the communities who are directly impacted and we help them address questions that they bring to us. So those are the coolest and most interesting studies, and drug checking is one of them that we will talk about today. If you go to the next slide, please. So what we're going to show you today is not a research study. Our approach is different. We like to think of ourselves as scientists in service to community. And the work that we're doing with analyzing and keeping track of the street drug supply is a public service of a public university. And that's our primary goal. So if you go to the next slide. We all know that street drugs change constantly, and we're still losing too many people we love to drug-related problems. But we don't find out what's in street drugs until it's too late, until only when people are dead or they're arrested. There's no opportunity for redemption. There's no opportunity for recovery or prevention. So what we do is, if you go to the next slide, in our campus at Chapel Hill, we monitor street drugs as a public service. Next slide. Our original kind of inspiration for this came from when we were requested by a local syringe service program that had obtained a smaller drug checking machine, whether we could help them with some lab services to back up what they were doing. And I was sitting in a car during the COVID period. It was a COVID PCR kit, and it was a vial with a liquid in it and some swabs. And I thought, hey, if I can stick this up my nose, get results the next day with the virus, like why can't we do that for a drug? And that's initially kind of what got us going. So we designed these kits. There's a lot of design thinking that went into this, but if you go to the next slide, the kits are, we provided them to about 170 programs from around the country we've sent kits to. And these are harm reduction programs, health departments, clinics, hospitals, drug user unions, EMS, and there's a vial in there. They put the substance in that vial and send it back to us for analysis at the lab. If you go to the next slide, the samples can be collected in 4 different ways. So we can get like a scoop of the powder, which is tiny, like less than a grain of rice. If if that drug sample has already been used, a wet swab on the inside of the vial can work. A piece of a fake pill, and a used cotton. So cotton is just a little plug of cotton that's used to filter street drugs in solution before injection. So those are the 4 main ways. This is what it looks like in practice. This is Ainsley Bryce at Holler Harm Reduction in Western Appalachia, North Carolina, who works closely with us. And it's really important that the samples are given to us voluntarily and that the tests are anonymous. And so the results are provided back to people anonymously. So because the samples have to be given voluntarily, we don't work with law enforcement because those samples are taken without without consent. If you go to the next slide, are the data that's being produced. So there's a card because the molecules don't tell the whole story, right? And so this card is our kind of very pared down data collection instrument. And there's, in addition to kind of the physical description of the drug, there's also information on if it was involved in an overdose and what sensations were experienced with that particular sample. The samples come to campus via FedEx, and we are in compliance with all the postal laws and the controlled substances laws and have all the licenses and everything that is required. And once the samples arrive, we unbox them and enter the data. The If you just keep going on the slides, the samples are we use GCMS, so gas chromatography mass spectrometry, and so this we used untargeted search. So if there is a substance in there that's detectable by GC, we will track it down and find it. So we're not biasing ourselves to like a limited panel or like set of substances. We're looking for whatever is likely to be psychoactive in that substance. If you go on with the slides, this is kind of what it looks like on a day-to-day basis. So far, we've analyzed 7,200 drug samples from 141 programs have returned kits to us, and that's in 36 states and 212 counties across the U.S. So far, we've detected 326 unique substances in the street drug supply. And, I think on a casual level, any of us can name probably a dozen, 2 dozen, like major drugs, right? But what we see in the drug supply is way more variable. And 326 seems like a big number, but we talked to our colleagues in Canada who have been doing this work for a lot longer, and their numbers are like up at 800 or 1,000 unique substances in the drug supply. So each one of these molecules has its own character, has its own kind of effect on the human body. And differentiating between them is really important if we're trying to avoid specific health harms. And Anuja will go into that more in just a moment. Most importantly, we provide results back to the individuals. There's a QR code with a sample ID. People can look up the results that they've donated to find out what's in them. There's no login required, no account that needs to be created. This is meant to be very low threshold. So we've kind of like what we're trying to do is build a not surveillance, like a data system that is first and foremost there to help individuals. So instead of thinking of it as data first, it's how do we help people first, and if there is data that comes out of it, then we can use it for other purposes. But first and foremost, our goal is to help people make better decisions about what they put in their bodies. And because we're taking an undirected approach, what's been really cool as a technologist, is watching how people take a new technology that's been liberated and use it and apply it in their own circumstances to ask questions that are relevant to them. They don't have to tell us the answers, but but it's been really cool. And this is the same as it is with technology adoption and other parts of science, right? So some examples are a group in Texas wanted to know if there was fentanyl in the black tar heroin there. Um, we often get samples from people whose partners who have died of an overdose and are waiting for an autopsy result. And that process can take 8 months or a year, and they want some closure earlier. So, what killed my husband is a question that we sometimes have to help people understand. There's even questions that address like misinformation or questionable information on the street. So there was a group in Nevada who was addressing a street rumor of like, can black lights like be used to identify these two particular contaminants in the drug supply? And we were like, okay, chemically, maybe theoretically, there's these possibilities. So they were able to look at those samples themselves, send us those samples, and then we analyzed them without knowing which ones were fluorescing which colors, and they were able to answer that question for themselves. And they didn't actually tell us what the answer to that question is because it's not directly relevant to the science and work we do. It doesn't affect how we can protect ourselves. So what happens in Nevada at that point in time with their drug supply is their information, and we're happy to provide this as a service. So that's kind of how we've intentionally designed this to give up certain aspects of control that other science and research tends to hold on to, but in doing so have really opened up the possibilities of people like doing citizen science, people answering questions that are important to them. So at the end of the day, we're all about helping people make better decisions about what to put in their bodies. Once we get the results, we are also able to if we find something that's a new substance that has never been tested in humans before, we have a lot of colleagues here at UNC who are lab chemists who we can provide a sample to, and they can tell us, like, this is the brain receptors it binds to, here's what we would predict based on the structure, and other quick information that can be used to kind of assess how grave a danger or what kind of physical harm we might be seeing from that particular substance. So here's an example of a real substance of a real sample that we analyzed. This is from Asheboro in Central North Carolina, and it's a messy brew of 16 substances. There's human medications like Tylenol, but also melatonin, which is a little weird. I'm not sure why that's in there. But there's also veterinary medications and numbing agents and stimulants. There's all sorts of kind of, so this one looks like it was heroin combined with fentanyl, and there's a lot of organic material in there. So something like noscapine, like the next question is like, what does that mean and how do we make sense of it?
Great. Thank you, Nab, for that awesome introduction and how the Street Drug Analysis Lab brings in the samples and then breaks them down by the GMS system to different substances. So, now that we have effectively turned these complicated samples into a slew of substances, how can we change that into informative action for our communities that can lead to, more safer interactions with these drugs? And this leads to our first problem statement. So when substances come into our GCMS system and are returned, community members are often left to search for these chemical names up on a search engine like Google. And in the best case scenario, these results are mysterious and arcane, but in the worst case, they can be misleading, confusing, and can leave community members often frustrated. So as a use case example, let's take noscapine, which Nav was talking about the last slide. And what's read out from Google is noscapine is an isoquoline, non-narcotic alkaloid derived from the opium poppy, somniferum with mild analgesic, anti-tussive, potential anti-neoplastic activities, which is such a mouthful. So unless you're a drug expert like Nav is, you're not probably going to know what any of this means. And more importantly, I'm not getting any, harm reduction specific information so I can, figure out how to interact with this drug more safely. So further, not only is like complicated information coming up, but just too much information is coming up, and people don't have the time to sift through that information for harm reduction specific. Okay, so what do we need instead? we need harm reduction specific translations. We need information that will support users to have safer interactions with drugs. So our solution is what we're calling the chemical dictionary, which is a database housed in Smartsheet and curated by our team at the Street Drug Analysis Lab. And I'll go into what the dictionary contains in a minute, but let's first finish up this use case of noscapine. So, Google is outputting these mysterious names, and what our chemical dictionary will do instead would output something like it lacks significant hypnotic, euphoric, or analgesic effects, affording it with very low addictive potential. So instead of searching up noscapine on Google and getting these mysterious results, users will instead get harm reduction specific information that is concise and that has been pre-vetted by our team of drug experts. So now that we can see what the chemical dictionary can do, what else is in it, and how did we create it? So the chemical dictionary is a database of 270 and counting unique substances, which are pulled directly from our samples. They contain a identifier key, which links to external databases that are outside of the harm reduction scope, like PubChem, which is hosted by the NIH, and CAS, which is hosted by the American Chemical Society. It also has things like substance synonyms, street drug vernacular, and pronunciation. But I think what the most important and interesting feature of our chemical dictionary that kind of sets it apart from more traditional substance taxonomies are the 6 words. And so here are some examples of what the 6 words can look like. Rotting fish smell, classic upper and caffeine pills, and bitter taste used in rubbing alcohol. So really like up-to-date, concise, situational information. So the chemical dictionary also contains a categorization process and a tagging process, which we'll talk about next. So tags are usually one word that describes the substance in their chemical, pharmacological, and street smarts and overall trending presence in the drug supply. So taking noscapine as an example, once again, these are the tags that our curators came up with. Things like plant grown, weak opioid, heroin impurity, established in the drug supply, opioid, and downer. So tagging allows for flexibility in defining the characteristics of the ever-changing definition of a substance. A drug's use is not static in the drug supply, and science is ever evolving. We want a dictionary that can adapt to those changing definitions. So this leads to kind of our final component of the chemical dictionary, which is categorizing these not so categorizable drugs. So on top of the ever-changing nature of these substances, substances are complex and they don't always fit neatly in these typical hard and fast boxes that we want to put them in. So for example, something can be a psychedelic, but also have a sedative tag, which seems like conflicting ideas. So for the purposes, though, of data visualization and generally making our data set more accessible, we want to try to cut through a bit of that complexity and categorize these substances to draw more general conclusions and to kind of open it up to a wider audience. So we created these 7 boxes that I think generally do a pretty good job of separating these substances into classes. But as I said, there is that nuance in defining a substance. So that's why our automation process was overseen by a team of experts to double check that this automation is correct. So when we do that on noscapine, we see that it ends up in the opioid bucket. So now that we have the context for our chemical dictionary, let's talk about how we can serve this information in the most effective and inviting format for our audience. Our first mission is to help the customers we serve, the ones who are sending the samples in. These are frontline public health programs, often without robust data analysis capabilities. We make our data sets available on GitHub without GUAs or additional permissions, but still programs have basic questions they can't answer. So our solution to this is to make live reports of each program's data to help them benchmark what is normal for their own drug supply in their local area. So that way they can add their own local context, which we don't necessarily need to know about, and provide actionable advice for people who use drugs to protect their own health. So, examples of this could be the top 10 substances identified. And if you see this description column, it's pulled, from the 6 words of the chemical dictionary. There's also, samples analyzed per month. We also did the combination of substances that led to overdoses. And so these live reports were done by an amazing student researcher and fellow MPH student, Bryn, with the help of NAB over this past summer. And it used DeepNote, which is NAB and I's favorite application to use. It's incredible. So if you don't want to get your hands dirty actually writing code, it's a really easy way to run data analysis. So now that we've kind of set the stage of the chemical dictionary and also made these live reports for our one customer, which is our community programs, we want to zoom out even further and show something that Dr. Green from Brown University talks about, which is the drug supply is not this massive, lethal, heaving beast. It's something that we're coexisting with. So our vision is to make the street drug supply seem knowable and not this overwhelming beast. We want to serve a broad audience, and we want a conversation starter. So right now, our audience mostly contains, communities that use drugs and community partners, but also we want to target, drug experts, researchers, clinicians, people who work with patients on a day-to-day basis. And even further out from that, we want to reach the general public and media outlets. And we want something that's just like a conversation starter, something that will draw people in. So our next project after creating the live reports is to create something that is approachable, effective, and interesting. So how are we going to do that? So let's first talk about what exactly we want to show, and then we can figure out how we want to show it. So our data is inherently hierarchical, as shown in this data, this hierarchy tree. So as you can see on the top, there's substances, and then the 7 classes of chemicals, and then all of the 270 and counting substances that kind of go into all these bins. And I've only put 2 in each for brevity's sake. But we also want to show what our lab is doing, which is showing the co-occurring nature of these substances in samples. So for example, we want to be able to show that fentanyl occurs, these many times in our database and is found with these samples, which are in these classes. So now that we know what we want to show, how do we want to show it? So there are several types of hierarchical visualizations that various packages allow us to create. One is the tree diagram, which may not be the most effective because we have 7 classes and we have a lot of substances and it might get pretty crowded. Then there's circle packing or enclosure diagrams, which is eye-catching but doesn't show the co-occurring nature of our substances. And then there's tidy trees, which are interesting. And so, we could maybe show the classes through the colors and maybe the co-occurring nature with these lines. So let's see another example of a tidy tree. So when a tidy tree only has one level, as seen on the left, it's called a dendrogram. And dendrograms are interesting because they can be reformatted to take up space more efficiently, and therefore you'll have more space to add, more information like the co-occurring nature and maybe the frequency of substances involved in our database. So on the left, you can see that this like Cartesian coordinate system, where in our case, all like the 270 substances would be kind of stacked on top of each other, which would be quite a long list of a long diagram. So instead, to kind of take up space more efficiently, we used polar coordinates and created a radial diagram that kind of wrapped that around itself. So this is what we want to create. So like now that we know, how do we actually create something like this using the chemical dictionary and the samples? So as you can see, this is the data processing pipeline. After drugs come into our central kind of our content management system, our CMS, it'll go into the chemical dictionary on Smartsheet, where things will be tagged and categorized, as I was mentioning before. And then from there, it will go to a Python IDE using an API automatically. And things will be converted into a JSON format, a flat JSON format. From there, things will be fed into a HTML script where D3 can be called using JavaScript and convert that flat JSON into a hierarchical format, which is what the tree diagram would show. And then that would be able to be rendered into our HTML domain. And that's what we did with our data. So once this process is completed, as you can see, this is the hierarchical edge bundling diagram, or the dendrogram that we created. So as you can see, there's a lot of components here. So one of them is the substances, the 270 substances that kind of wrap around the circle. Each one has a corresponding node to it, which the size of that node will show the frequency of that substance found in our database. And the links will show, the lines will show the co-occurrences of those two linked substances together. And we also split things up by classes. So in the center here, we have the opioid class. On the top right and left, we have synthetic cannabinoids and psychedelics. On the right, we have sedatives and stimulants. So the sky is really the limit with this. We plan on creating a poster, which is in the works right now, with the help of our amazing graphic designer, Britton. And you know, we hope to show more of like the nuances of this visualization and what's kind of getting like to get a little deeper and tell the stories of a link or two or specific substances inside the poster. And we hope to send this to community programs to be hung up in their offices and get a conversation to start and maybe get people interested in looking into what this is further. Brendan's also been doing an amazing job of kind of reformatting our visualization and adding awesome graphics like what you see on the bottom left. So D3 is really cool because it's dynamic and it allows for interactive user experiences. So our visualization could be hosted on our website and turned into interactive, like the nodes and the links can be clickable and can maybe link users to more information. And we're hoping that it's going to be like a central location for our audience to come in and dive deeper into the data nuances. So some key takeaways here is that, the drug supply is vast, but it is knowable. And through data techniques, we can shorten that knowledge gap and provide community partners with effective and real-time solutions. And art and data visualization can draw, all kinds of audience members into the conversation, which is what we want. So thank you so much to our incredible chemical dictionary team, Nate, Nav, Meredith, and Aaron. They've been working hard on curating the chemical dictionary for the last 6 months. And it's pretty amazing what this chemical dictionary is going to be able to do for communities. So we wholeheartedly thank you for spending your time doing this. And I think, Nav, you have one more slide here.
Yes, I just wanted to thank everybody on our team, especially Colin, who has been working very closely with Anuja as our mentor for kind of bringing the real world experience into the data and kind of how we tell the story. And Britton, who's on the line right now too, is our awesome graphic designer. And as you can see, like there's like stuff you can do in code, and sometimes things that even when you think it looks pretty, Britton finds ways to make it even prettier by usually by making it simpler and kind of focusing on the core message. So we're looking forward to seeing where the collaboration between Colin, Nuja, and Britton will take this to make this into posters that we'll hopefully finalize and get out to you all this to all our frontline programs this fall. So our newsletter is kind of going to be our way to break ourselves of some of our social media dependencies and kind of try to control our own narrative a bit. And if you want to sign up for that, we'll be posting all the details of this as we go along. So thank you to our awesome team, especially our lab folks, Erin, Jalis, and others who are the ones who do the physical chemistry, and Shay and Ilyana and Colin and others who are packing the kits and doing the mailings every day. This is a lot of manual labor that we appreciate everybody deeply for. So thank you very much. We're happy to go to your questions.
That was fantastic, guys. Thanks so much. So in just a second, we will be pulling up the Easy Retro board, which is our Q&A platform for this event. And I just want to walk you all through, pretty quickly. You've got the link in the chat. Open that up, and I want to walk you through how to add ideas. So you simply click on the plus sign in the relevant column and type in your idea or question. And then when you're finished, you hit the blue box with a thin white check mark to save within the same field. And then your idea will appear on the screen for the public's reaction. You can also comment on other ideas and questions, also known as upvoting, by using the thumbs up icon, and you can comment on an idea using the quote bubble icon. So feel free to use all of those tools. We are really eager to hear about the awesome questions you're going to have for Anuja and Nab. I'll let you guys take it away right now.
Great, thank you. So the chemical dictionary is something that we will fully be making it all public and open source. So just give us a couple more weeks to work out the kinks, and we would like it to be a collaborative space where if people can say something better in 6 words, then great, please tell us and we'll change it up. We're also trying to make sure that it's available and it'll be updated on a weekly basis. But we would also like to make sure that there's APIs and other connections that are easy to do into it, and that it's also canonical, and that there's a kind of responsibility and pedigree of when we change things and have that be historically available. So those are all our kind of design requirements when it comes to how we release that, but we will be doing that. So if you sign up for our newsletter, we'll definitely have the information going out in there soon.
Well, I can do one. So for the data visualization, what softwares did we use most? As I mentioned, Nav and I love to use DeepNote, which is a Gen AI software. It's a great tool. And then to create the visualization, I used D3, which is a package in JavaScript. Anything to add, Nav, on that note?
Nope. We have a convoluted pipeline now that is getting simpler, but the main website itself is a Squarespace website, and we were trying to get this service up and running really quickly. So it's actually just like a shopping portal, and each result is a different product. So we were really afraid that Squarespace was going to shut us down because it looked like we were literally selling heroin, but there's no kind of transaction or cart or other kind of functionality there. So that was the kind of expedient way to get the data out there with like existing off-the-shelf tools. And yes, we do record what substance the donor, thinks it is, and there is, so one of the things that Bryn has been working on this summer is figuring out a way to visualize the concordance and discordance with that, which sounds relatively easy, but in practice is hard because the same drug can be called different things in different parts of the country. And then the same molecule may have, it may be in like the same family, but just not named specifically. And we're like, really, we don't expect the donors to know the difference between like, p-fluorophenols and, p furanylphenol or something, like just like there's little nuances in there that we want to make sure that we're accounting for to not like discount someone's knowledge of what they thought it was. And so that's where the chemical dictionary and the categorization really helps us get that metadata that will then make that work a lot easier. So we'll be rolling out those program level views, live reports, and summaries this fall as well. So we're just kind of going through and kicking the tires to make sure it all works and works fast, and is like optimized for browse or for handheld devices and things like that. But that's coming soon. Let's see. There's a question about why not use a co-occurrence matrix. So, Anuja, do you want to talk about like data structure?
Yeah, sure. Well, so I mean, I think it has to do with kind of what we wanted to show, like the different components of what we wanted to show in our visualization. Um, and so like a matrix is, this like almost like a heat map of like the different like co-occurrence or like what are found. And I think the problem with a matrix is that it's if, for example, we have like an infinite number of substances, that heat map, that matrix will be so large, and not be very like readable to the human eye. And so I think we wanted this kind of like circular, smaller visualization that is just a good, like efficient use of space. And that's kind of why we settled on that structure. But matrices are great for smaller, like data structures and smaller like amounts of data. So great comment.
Staying on the data structure type question. So there's one that is, would you consider integrating time component into the viz and capture trends over time? And yes, definitely. We want to, and this is like where we're doing things a little bit differently than in traditional public health, where those visualizations and timelines would be kind of for public health authorities, for epidemiologists, for people like with degrees and kind of fanciness, right? But when we think about who needs to know that information, it's really the programs, because they will have so much more information about what has changed in their local drug supply, and and the different suppliers, different law enforcement actions, whatever it is, and they actually are the ones who have the context to interpret the ups and downs in a timeline, whereas kind of we in like a more removed public health setting, like at a university, aren't the ones who have that localized knowledge. So we are intentionally prioritizing timelines for as a way for to give back to programs, but not necessarily something that is a priority for us to like share broadly in the public health arena. Because again, this is meant to be directly a service for people who use drugs, for people who care for people who use drugs, and their needs are more paramount than our data hunger and our data greed sometimes.
There's a, oh yeah, go ahead.
Go ahead, go ahead.
I was just about to put this one for you. Is there a way to get data of specific orgs or to get an alert when my org's results are up?
Oh, I know so many organizations ask for this. We are working on getting that up. It is surprisingly complicated to do that. It is on our software developers, like we're going to think about this very soon list. So we hear you, we appreciate the feedback, and we understand it's a pain to check for results. So give us a little bit. we're working on it. We'll find a solution. Twilio and other kind of automated text message and email service integrations. Yeah. Some things are easy, some things are not, right? How did you develop a relationship with your local graphic designer is a question I love. And I think we had someone from a designer from within UNC also post a useful comment in the board. But Britain has been working with us for almost 5 years. And the first pretty much year of our engagement, we did a deep dive into looking at how how drug war imagery how drugs are portrayed in news media and public health. And we found that a lot of times it was very sensationalized, a lot of like spoons and syringes, and really like dehumanized people, and like were, these color palettes that were really intended to make you see people in a bad light. And we wanted to make bring the humanity back. So Britton and I spent like that first year just like thinking through these concepts, coming up with the right design aesthetic for all our work. And it's been a really, really fruitful collaboration where I can chicken scratch and scrawl my little idea with like little figurines, and he can say, he'll make it into something that's really a much better communication tool. So we deeply appreciate that engagement that we've had. But it took a lot of work up front to um, get through the public health concepts and really, be respectful to the people we serve. So some of the questions on chemistry and public health. So nitazines, yes, we have seen nitazines in North Carolina, but 2C we have not seen in North Carolina yet. We have seen some, I think, on the West Coast, but it's possible it'll show up here. But they're usually 2C is interesting because it's not like it's a combination of two or three different drugs, often centered around ketamine. But what is called 2C will also change from festival to festival or from month to month. And so that exact constellation of chemicals that makes up 2C might not be consistent. So it is something that we're keeping an eye on. It's not like on our the top of our kind of concern list at this point. New substances, yeah, someone so whoever posted this question really has been following what's been going on this summer. Bis 2244, what is it? Tetramethyl 4-piperidine sabacate is a plastic. It's an additive that's put into plastics to make it UV resistant, and is also a fragrance in certain kinds of Glade candles. And earlier this summer, mid-June, two programs in Los Angeles and in Portland, Oregon, that are using point-of-care drug checking equipment identified that as a potential contaminant in the drug supply. And then we were able to confirm exactly what that was. But it's a plastic smelling compound that's pretty noxious. And usually those kinds of things come and go and disappear out of the supply. But this one seems to be sticking around for a bit, and we've detected it in about 11 states. So not exactly sure why that one's showing up, but it is, and it's causing some pretty nasty reactions in terms of blurred vision and like really bad coughs and throat burns and things like that. So not so happy about that one. Okay, so have we collaborated with other labs? Yes. And in fact, our lead chemist, Erin Tracy, or leads up or volunteers her time to lead up a national and somewhat international consortium of the other labs who are in this space of doing public health street drug analysis. And so we had what was it, like 8 or 10 of the labs around the country on our campus earlier this summer to coordinate methods and figure out how all the super chemical nerdy questions of like how much solvent are you adding to this and what's your runtime on that. And it was really great to have them. So yes, we are super collaborative, and we also work with the point of care drug checking programs that are frontline who are doing this. And the smaller FTIR machines can get you results back in less than 10 minutes. So we think that's like first and foremost, like where the investment really needs to go to make this a broader public health paradigm. And we support organizations like Remedy Alliance and others to create a training curriculum and standards for getting those programs up and running. There's about 60 of those machines out there right now, and we're hoping that will grow, but we're happy to provide the backup laboratory services for them. So any of the other ones, Anuja, that you wanna?
I'm seeing there's an interesting one. I see you capture the geographical location down to the county. Do you plan on comparing locales with different with similar chemical profiles? Do you think that kind of relates to Bryn's project at all?
It does, but I mean, I think before I started doing this, I was a bit naive about drugs, even though I'd been studying them for 20 years. I mean, what we see is that um, the same stamp, the same product sold on the same day in the same city can look completely different. And so before we start drawing a lot of like inference into how things are moving and kind of population level data like we would in normal epidemiology, I think we really need to understand like what within person variation is like, and then kind of really let the programs lead. I mean, our goal is to not be answering questions that we generate, but rather help the programs answer the questions that they're interested in. And we're here to be a scientific service. And so, yeah, we have lots of questions that we could ask the data, but really, I mean, we're here to serve the community first.
I can do one that's in the miscellaneous category of different data courses at UNC that students can take. I think there's a new data science and society department that just started this year that has a lot of really good courses. There's a great data mining course that gets into looking at wrangling messy data, which is usually how data is in the real world. It's never kind of perfectly cleaned. So definitely like look outside of kind of the BIOS department for kind of those like big data courses. And message me if you have any other questions about that.
All right, folks. Do we want to take one more question, or are we ready to move to our wrap up? Lots of appreciation and thanks. So amazing. All right. Well, thanks so much, you guys. I do want to draw everyone's attention. If you are still here, I'd love to draw your attention to the chat. My colleague Jay Shree has noted that we're interested in hearing about the data skills needs for our attendees today who are working in public health or community engagement or some mixture of those fields. If you could take a second to fill out our data needs assessment survey, we would love to be thinking about you guys as we plan our 2025 programming. So take a minute to fill that out, pass it around to your friends. We would love to know what your needs are. And next slide. I am super excited to share some details and highlights from our fall programming calendar with you guys. We have a really great event coming up on September 11th at 5 p.m. Eastern, featuring Trace McMichael from the Kennedy Center, who's going to be speaking to us about social impact civic alliances that are taking place at Kennedy Center and his management role in that. Next slide, please. We also have Dr. Krista Arangala from Elon University, who is going to be presenting a little bit of information about how Elon is developing a coordinating hub for data competency. This is part of their effort to become an accredited institution in their data nexus program, help them become accredited for their data science programs. And next up, we have our event, AI for non-coders. This is great for folks who might be working in a field like public health or local government, where they are expected to know a little bit about AI, but need more information about the technical aspects of it. So this class is geared towards folks who are not the technical people on your staff, but who might need to have some expertise or some awareness about how some of these tools function. And I think we have one more slide. Yes, our amazing, super excited about our annual data science career panel this year. We have some great speakers from IBM Research, Accenture, and Uplift Labs. And Uthra Ramunajam is our moderator for the event from Liberty Mutual Insurance. So do make sure you take a second to sign up for that, or tell your friends in your life who would be who are looking for careers or internships or those sorts of things. We'd love to have attendance from all over the country once again. And I think we have another slide just to say thank you so much for joining us today. You guys are amazing. Those of you who have stuck around, we really appreciate it. We are always interested in identifying new DataBytes speakers, so feel free to fill out the interest form or just shoot me an email if you are interested in serving as a speaker or know someone who should. And we do encourage you to stay connected with all of the activity that's going on at the Opioid Data Lab by signing up for their newsletter at opioiddatalab.ghost.io. So thanks so much, everyone. This was a great kickoff to our afternoon. Really appreciate you guys being here. As always, Nabarun, lovely to see you. Anuja, great to meet you. And we look forward to hearing from you guys in the future. All right, take care, everyone.